Hi everyone, and welcome to our video on PCR. This is part of our Module 6 Part 3 series looking at manipulating genomes. And in this video, we're going to be having a look at specification reference 6.1.3d, the principles of the polymerase chain reaction and its application in DNA analysis. Now, the first thing we need to kind of bear in mind is why PCR actually came about. Why do we need this? And when we're thinking about that DNA profiling from our previous video, we were talking about how we could use DNA from a variety of sources and then work out basically a little profile of who it came from. But in order to do that, we need a sufficient quantity of DNA. Now, the downside is that in a lot of cases, particularly criminal activity, generally, criminals aren't going to leave large quantities of DNA at the crime scene unless they are truly terrible at what they're doing. So we may only have a very low quantity of DNA available from a crime scene to utilise in any DNA profiling. So we developed this process called the polymerase chain reaction, or PCR, and what we do is we amplify the amount of DNA. So it's a way for us to take a very small quantity from a crime scene, for example, and then make large amounts of it, which we can then utilise in things like the DNA profiling. In terms of how this works, when we first developed PCR, then it was actually really quite slow. Because what we found is that in order for the DNA polymerase to work, and DNA polymerase, if we think back to our Module 2 work, is the enzyme that is going to basically create new strands of DNA from a template, that had to be kept at 35 degrees Celsius. So 35 degrees is obviously not a very high temperature, but we couldn't go higher because otherwise we'd denature our enzyme. So we were a little bit stuck. We had this very slow process amplifying our DNA just because of the polymerase we were using. We solved that by actually discovering a different form of polymerase, and this is something called Taq polymerase. Now, the reason it's called Taq polymerase is because it comes from an organism called Thermophilus aquaticus. So Taq is the capital T and then the AQ, so Taq polymerase. And this Thermophilus aquaticus is actually an organism that lives in incredibly high temperatures. It's a thermophilic bacterium. So thermophilic, if we think about that word, thermo is obviously to do with heat and philic is the liking. So a thermophilic bacteria is one that really does like heat. It lives in these areas that are really quite hot, and therefore the enzyme inside them, the Taq polymerase, is able to work at much higher temperatures. So all of a sudden we've removed that barrier to being able to use higher temperatures in this process of amplifying our DNA, which means we can speed it up. So how does it happen then? Well, we've got these three key stages: denaturation, annealing, elongation. So the first stage, denaturation. We take our DNA and we're going to mix it with DNA nucleotides, some primers, magnesium ions, and the Taq polymerase. And what we're going to do is heat that double-stranded DNA to 95 degrees Celsius. And when we do that, it breaks the hydrogen bonds that hold the two strands together in our double helix. So obviously we've got hydrogen bonds between our complementary base pairs. When we heat it to 95 degrees Celsius, we break those hydrogen bonds and therefore create single-stranded DNA. Step 2, the annealing part. This is where we decrease the temperature down to 50-60 degrees, and that means that the primers can then anneal to the ends of that single-stranded DNA. So what we end up with is our bit of DNA, and then we've just got this little bit where the primer has joined, and that makes a short section of double-stranded DNA. And that is where our enzyme, the Taq polymerase, is going to bind. Third stage, the elongation. Temperature then gets increased to 72 degrees Celsius for at least a minute, and that is the optimum temperature for the Taq polymerase to build those complementary DNA strands. And they always run 5' to 3' direction. Now, obviously, we're not just going to do this once. We're actually going to cycle through this over and over again through several cycles. And every time that we go through a cycle, we are going to double the amount of DNA that is present. Because every time we heat it up to that 95 degrees, we're going to break apart all those double-stranded bits of DNA into single-stranded templates. And then as we cool it down, they're going to become, obviously, bound with the primer, and then our polymerase is going to copy it as we warm it up once more. So what we do is we repeat that process for several cycles, and we get this exponential increase in the quantity of DNA available. The last thing we really need to understand as far as PCR goes is when we're going to use it. So what are the applications for it? Obviously, we've been talking about the forensic side of things, using small amounts of DNA from crime scenes, amplifying it up so that we can run those DNA profiles. But that's not the only use. We will also use it in terms of organ transplants. So if we're going to do an organ transplant, we need to make sure that the donor and the recipient have the same tissue type. If we don't do that, the organ that we're transplanting into our recipient could very well end up being rejected by their body, because what it will do is very easily identify it as foreign, and as a result of that, the immune system is going to attack it and basically kill the organ. So by using tissue typing, we can match donors and recipients before that transplant takes place, reducing the risk of rejection and also reducing the potential wastage of very valuable organs. We can also use it in research. So we can obviously utilize DNA from an extinct source where clearly there's not a huge quantity left. If an animal is extinct or a plant's extinct, we're not going to have massive reserves of their DNA available to us. So using PCR, we can amplify it up, and then we can look at that in things like the evolutionary links and trying to piece together those evolutionary family trees. We can also use it in terms of identifying viral infections. And this was something that you are all probably highly familiar with based on the fact that you all lived through a global pandemic. So we have got, obviously, good old COVID-19 coming back as a good example here. When people were suspected of having COVID-19 back in good old 2020 and 2021, then you shoved that lovely swab up your nose and down your throat, and then you handed it over to a person wearing a lovely full kit and in a little baggie. They then sent you a message saying, yep, you've got COVID-19, or no, you don't. And what they're doing there is they're using that small amount of viral DNA that would be on that swab, amplifying it up so it's then detectable, and that means we can identify infected people. So anytime that we're trying to do this sort of viral infection work, where we're trying to work out who's got a particular condition and therefore at risk of spreading it to others, we want to be able to find that from small quantities of the viral DNA that may be present. We can also use it in these epidemiology studies, and epidemiology is looking at the spread of diseases. So again, we can use COVID-19 as a good example here in the fact that we were taking that DNA from those swab tests, and then we were able to carry out the tests to identify the different strains of COVID-19 that were present. And from that, we could work out where this new variant had popped up from and where it was spreading to. We could track which ones were of greater kind of risk to the population than others. So that means that we can monitor how it's spreading through a population, and we can identify when it's mutated. And obviously, if a particular mutation leads to a more risky variant, we can take different steps to obviously control that. We can also use it in detecting oncogenes. Now, oncogenes are basically the genes linked to cancer. So wherever we see onco, that bit there is relating to cancer. So oncology wards are your cancer wards in hospital. So the oncogenes are the ones that are the genes linked into certain types of cancer developing. Think things like your BRCA gene for breast cancer, etc. And if we can obviously detect those, we can give people better advice about steps they could take to reduce their risk of suffering from obviously quite serious forms of cancer later. And the last one, we can actually detect mutations as well. So when we're having a look at some of the kind of things that we can now do when it comes to reproduction in humans, when we're thinking about people who may be at risk of certain conditions, etc., we can carry out screening on the little embryos before they're implanted in that process of IVF. Now, what that's called is pre-implantation genetic screening. And what they're then going to be able to do from that is they'll be able to identify which of those embryos carries the particular genetic condition that we're mindful of, and then that won't be implanted. They can literally select the embryos that don't have that particular genetic trait. We can also do a similar sort of thing, but taking blood from mother during pregnancy and then identifying certain conditions from the actual fetal cells that are present within the blood there. But all of these things are going to give us a very small quantity of DNA to start, which won't be enough for us to do all of our tests on it. So we need to amplify it first of all. So we use our PCR process with that Taq polymerase to give us a larger quantity that we can then put through the process of DNA fingerprinting or DNA profiling. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next videos are uploaded, and of course head on over to the website where you can find a range of other resources to help you in your A-level biology studies.